This is a production of Cornell University.
Good afternoon. I'm going to be talking about soil health in India, and don't worry if you know nothing about India, because I will cover some of that as I go through this presentation. I also intersperse this with quotes, both in the anglicized version of Hindi and the English translation, just to tell you what Indian people think about soil as you go through. So it's a quick outline to my presentation. Like I said, I'll give you the background of India as well as soil health in general, the way I'm looking at it, and the comprehensive assessment of soil health, which was developed here at Cornell. So actually, that is Cornell up there. Go through some of the objectives, the methodology for my 3 objectives, and I'll end with some preliminary results, most of which is all of which is on my objective 1 right now, so I don't have too much to present to you. Okay. Quick background, this is India, if you haven't seen a map of India yet. The yellow is arable land, so a lot of yellows, a lot of forest, green, and the total area of 328 million hectares of the country, of which 60% is agricultural land. Of that 60%, about another 60% is rain-fed agriculture. So most of Indian agriculture can assume is rain-fed, is smallholder farms, less than 2 hectares. And yet, despite all that, India has a pretty much equal export-import when it comes to agricultural produce. So there's some stuff that we import, others that we export, but as a net, we are pretty food self-sufficient. So as a quick this thing into Indian cropping systems, we actually have 2 to 3 cropping systems a year, unlike the US. So you have the Kharif cropping season, which is usually in the monsoons across the country, so June to October. And there you'll have rice, obviously, again, monsoons, corn, cotton, all of which is grown in this season. Then you have a dry season crop, which is Rabi. And then that you have wheat, barley, mustard, a lot of the pulses are grown in this time. And some farmers will try to squeeze in a summer crop in between March to June, and that's usually melons and gourds and other short season crops. Okay, so getting into the meat of it, soils. This is a soil map of India. The yellow is alpha soils, so a lot of alpha soils. The west has a lot of vertisols as well. But where I'm focused is up over here, which is the northeast. And there's mostly inceptisols and entisols. Looking even further at an actual map of the state, which is Bihar. This is northeast corner, so you have Nepal up here, Bangladesh. There's the state of Jharkhand below, where someone else in the lab, Phil Frost, has also done a lot of research. And this state, the interesting thing is it's bifurcated right in the middle by the Ganga. So the Ganga cuts through the state, and generally how agriculture works there is these are mostly young alluvial soils, and a lot of nutrients that are coming with the river generally flood the local area slightly, and then there's a renewal of nutrients almost every year because of this. It also leads to excessive flooding, especially in recent years. So that's definitely something that causes a lot of risk in agriculture in that area. Where we are is this little, right above the river, this little purple area, which is actually calcareous soils. So a lot of the soils that I'm working with are calcareous as well. Okay, here's a better picture of the state itself, Bihar. The total population of this state is 99 million. So that's more than a lot of countries in Europe, including the UK, Germany, France. All of them are smaller than that. And close to 80% of this 99 million all work in agriculture. So Bihar is inherently rural, and on a lot of developmental indexes in India falls towards the lower side of the categories. They mostly grow rice, wheat, and maize. There is a lot of vegetables that are produced in the country, even though they're not considered the primary production. And it also has the lowest farm income in the country, which I thought was interesting to mention. In general, in India, farming is not a lucrative career. It's about $95 a month you would get from farming. But in Bihar, it's less than half of the national average. So it's inherently a very poor state. So that's where soil health comes into play. soil health, generally also referred to soil quality by the NRCS, is defined as the capacity of the soil to function as a living ecosystem. In our case, that generally involves sustaining plants for food production. It has a number of functions that we try to define as important to that functioning of the soil. And the US especially, a lot of the way soil health has been seen is looking at the physical, chemical, and biological processes of soil. In India, it's slowly starting to be seen that way, but for a lot of government policy, soil health is still considered mostly just chemical processes. So the soil health card scheme, for example, which has been currently run by the government and is costing the government $86 million, looks at just the nitrogen, phosphorus, and potassium of farmer soils and micronutrients, and considers that a complete picture of soil health. So why soil health? I mean, a big reason is because India is going to face a soil crisis soon. And all of South Asia, what we're seeing is that yields have to come from increasing production because all of land that could possibly put under agriculture is already under agriculture. Yet a lot of the soils that we have currently are degraded, which is leading to either decreased or stagnating yields. So improving soil quality can be a really important way for us to improve those yields. And the reason I'm using the comprehensive assessment of soil health to look at this is it was developed at Cornell. It's been tested over the years in multiple different scenarios. It's a very comprehensive approach that looks at the physical, chemical, and biological processes of soil health. And it's been tested thoroughly. And the way, just as a conceptual view of how this came about, is we have soil services, say, for example, for production, which is what I'm really looking at. And that's a result of certain soil functions that the soil does, like plant growth. The function themselves is influenced by the soil processes that are leading to these functions. And since we can't measure soil processes, we're going to look at certain indicators that will give us an idea of how these processes work or how these processes are progressing. So that comes up with a soil health test that we've tried to create, which looks at all three different processes that fall under the three different factors. And I kind of want to point out that this is also kind of a feedback loop. So if you have a soil health test that gives you recommendations, you continuously test for this, your indicators get better. That is influencing your processes that will eventually influence your soil service as well. Okay, so the cash indicators, I'm going to get into what indicators that we're going to be measuring, but just as a quick aside, the way these cash indicators were selected was, one, the relevance of that specific indicator to the processes that we want to look at. How consistently can we reproduce it in the lab? How easy is it to sample? And what is the cost? So putting all that together, we came up with 15 tests. And I'll kind of go over all of them specifically. So there's the chemical tests, which is soil pH, organic matter, extractable P and K micronutrients. This is kind of commonly known, and it's what India normally does for the soil health card. And the biological tests are active carbon, soil respiration, and a protein test. And then for the physical characteristics, we look at aggregate stability, available water capacity, and surface and subsurface hardness. And as kind of an example, you have one of the final analyses that we do of a 10-page report. So this is the top page. You have your physical characteristics, your biological up over here, and then your chemical. And you have the value that you calculate in the lab, whatever the units are. And each of these values are then converted to a rating that I'll explain how we get in a little while, which is kind of on a sliding scale. So it goes from 0 to 100. Obviously, if everything is great, you get a little green rating. It goes up to 100. If for any reason your score is very low, it becomes maybe a red, and it's towards the lower side. And then eventually you have an overall score, which is an unweighted mean of all our indexes. And we also keep texture into mind because obviously all of these indicators are going to be affected by texture. So that kind of will change how our rating is taken. The rating you can have three different kinds of ratings. There's one which is more is better. So it's generally a sliding scale. Up over here is active carbon as an example. So more but the higher the value of active carbon, the better it is. There's some where lower is better. So surface hardness, for example. So obviously too hard of a soil is bad. And then there's some that's an optimum value. So pH, where you want it to be neither high nor low. And how we convert the raw score into an actual rating is we have a normative evaluation. So you have, you're comparing each sample that you analyze in lab to a larger population of samples. And all this population of samples has been like, you put up a histogram, you have all your samples in place. They're mostly all normally distributed. So you have a mean and a standard deviation that you get from that. And then you convert that to a cumulative normal distribution. One thing that's really important, all the samples that were used to define our current distribution function have all been taken in the US. And that is going to affect the rating, because as I may point out later, a lot of this like values that I've gotten in India, a lot of values that other people have gotten in India are all on the lower side of what you would consider here, what you would find here in the US. So it sways the function and it sways the rating, and that needs to be reanalyzed. Okay, getting into my objectives of what I want to do with this study. So I want to know what affects soil health, and I want to know how I measure it accurately and efficiently. So the first objective, which is, as I said, what I've really started and I've gotten the results on, is I want to look at the effect of different crop management techniques on soil health. What I will be getting into is assessing soil quality indicators using mid-infrared reflection spectroscopy, as well as developing an in-field test for some basic soil quality measurements. And I'll get into each of these a little in a second. The first objective, the study sites that I've mostly focused on so far, is first the Dr. Rajendra Prasad Central Agricultural University. So this is a university that I'm being hosted at, that I'm working at while in India. It's located in Pusa, Bihar, like I showed you in Samastipur. It was established in 1905, actually. So it was originally a British Imperial College for Agriculture, which after independence was renamed to the current Rajendra Prasad Central Agricultural University after the first president, in fact. And the second place that I've taken samples currently is at the Cereal Systems Initiative for South Asia. So this is under CISA, which is, this is under CIMMYT, which is the International Maize and Wheat Research Institute. They also work in collaboration with the Rice Research Institute and IFPRI, which is the Policy Research Institute, and I'm forgetting what F stands for at the moment. Okay, now that I've got the study sites, one of the first things that we had to do was actually set up a lab. And given, like I said, a lot of testing in India for soils is mostly on the chemical side, a lot of the equipment that I would need for the biological and physical indicators weren't present. So it was one of the first goals of the project. I spent like the first year creating this lab with the university. And it's a little tricky because you can't ship soils from India to the US. So we couldn't just analyze them over here. But it's also hard creating a lab there because there are some restrictions on just transferring foreign funds to India as well. So that was a very challenging process, but has been very fulfilling given that it's all come together now. Okay. So this is the methodology for how I'm going to get into my objectives. The first part, like I said, that I have started working on is, can we pick up significant effects of management techniques on from these long-term trials? I've already sampled trials at the university, at CISA. I'm looking at Borlaug Institute of South Asia, which also has a lot of long-term trials with a lot of legacy data. One of the things I'm hoping to do soon after is CISA is also collecting projects from around the state as well, and that analyzing those samples will also give me an idea of the differences between university plots and farmer fields. And so you can see which of these practices are actually being adopted, which of them are showing actual health benefits. And that kind of leads us into what will hopefully be building diverse and resilient systems off some of this data. And the experiments that I'm working on, and I'll come back to this as I get into my results, is mostly looking at cropping patterns, organic amendments, and so you'll notice zinc comes up twice. That's because a lot of these soils are zinc deficient. So that's one of the things that a lot of the university trials are studying. and also looking at 4 different scenarios at the CSAP plots, going from farmer fields to diversified systems. Okay, hopefully everyone's getting there with me. So the second objective that I'm looking at is assessing soil quality indicators using mid-infrared reflection spectroscopy. This has been done before on a number of indicators by a number of different scientists, and the results vary. There are certain indicators like clay content or carbon that have actually been predicted very well using reflection spectroscopy. There are a lot of indicators where we're not seeing any relation between the spectroscopy data and the actual indicator values. So I'm really interested in seeing, of the 15 indicators I've chosen, what are some of the conclusions we can make. And the way this works is you have a sample collection, you run reflection spectroscopy on all those samples, you analyze all the same samples in the lab, obviously, then you derive a model. And one of the common models used is a partial least squares regression model. And then so the next collection, now that you have a model set up, you can analyze 95% of your samples using the reflection spectroscopy, using mid-infrared. And then you have a model to derive your soil health indicators from that data. So this project, again, we're working with CISA and the university, and there are already about 1,000 samples collected that we will be working on. The third objective is working with MIT to develop a point of views, and I'm sorry if this is a little hard to read, I realize the light's on. So working with MIT to develop a point of views NPK sensor. And so they're currently working on a sensor that will measure just the nitrogen, phosphorus, and potassium. What we're going to be checking is an in-lab accuracy as well as an in-field accuracy. So like both once the sensor is made, how does that compare to soil tests in the lab? And if it does work in the lab, once we take it out in the field, how accurate or adaptable is it? And this is kind of from their data. What you have is a little sensor up over here. You have your extractant, soil. You mix the soil and the extractant and put the sensor in, and that will give you a value that you can then take to an NP or a K amount. In addition to that, in terms of like adding some information on the other characteristics, like the soil health information, Joseph Imaselli, who's sitting over here, has done some work on a best subset analysis. And the way that works is you're trying to find out, first, if you have a subset size of 1, what is the one indicator that will best predict your soil health value? Then what are the top 2 indicators that will predict your soil health value? And finally, the top 3 indicators that will predict your soil health value. And in the end, the top 3 indicators are all the same. It's organic matter, respiration, and active carbon. But interestingly, while in the US samples of 8,000 samples analyzed, active carbon is the best predictor of the soil health, the final soil health value, the mean. Whereas in India, based on Phil Frost's data, the organic matter is actually the top indicator of soil health. The reason we're interested in this is because active carbon is actually a test that's fairly easy to add to any in-field test. It can be done around the field itself. It's easy, it's inexpensive. So if we were to add one thing that would be able to also give us a soil health value along with the NPK data, active carbon is a very good candidate. So that's one of the reasons we're interested in having some more data from India. All right. Hopefully everyone's doing good so far. Okay. I'm gonna get into some of the preliminary results now. This has all just been recently analyzed. I've literally been getting data this week itself, so this is still very rough. But so the first experiment that we're looking at is, again, the effect of different cropping patterns and management practices on this yield and soil health. So they have 2 different crop rotations. You have rice, wheat, sorghum. That's more typical of the state. They're all grasses. And then the other is rice, mustard, moonbean. So moonbean is a legume. It's a little more of a diverse system. And on top of that, there are 4 subplots, so 4 treatments. You're looking at 10 kilograms zinc, so just zinc. Then you have 10 tons of farmyard manure, which I'm going to use as FYM. And then you have combinations. So 10 kilograms zinc, 5-ton farmyard manure, 10 kilograms zinc, 10-ton farmyard manure. And what we're going to find is, so this is our graph up over here. This first graph is looking at active carbon. The purple is the rice, wheat, sorghum. The blue is the rice, mustard, mung bean. And the way I've set this up is I've got farmyard manure up over here. There's no control, as you'll notice. So this is, the study was set up actually in 1985. So it's been a couple of decades, which is really great in terms of the amount of data we have available. But one of the issues is the way it was set up then and has continued means you can't really change it in between without losing some of that legacy data. So we've compromised by using a 10-ton FYM, that's 0 kilograms of zinc. Then you have zinc, then you have additions of FYM, so it goes up like that. And we have active carbon up here on the y-axis. And what you're seeing is, one, the average rice, wheat, sorghum active carbon is higher than for the legume rice, mustard, mung bean system. That's really interesting. It's also not a perfect comparison because these aren't randomized between systems. So you have system A, which has the 4 treatments randomized within, and then you have system B, which has the 4 treatments randomized within. So we can't fully tell, because the way this is set up, if the differences that we're seeing are due to the cropping systems or just spatial differences, without doing maybe some more further research, but that's some of the preliminary data that we're seeing. What we're also seeing is, in terms of the actual treatments, there's no differences in the rice, mustard, mung bean, which is the blue up over here. But we do see some slight, we do see differences between the rice, wheat, sorghum, where you can see the addition of farmyard manure is increasing the active carbon. What we are seeing is soil protein. We're seeing a similar trend, actually. So there's no difference, again, in the rice, mustard, mung bean, still in blue. But we are seeing the increase in the rice, wheat, sorghum up over here, which is interesting because, again, like I said, mung bean is a legume, but we're not seeing an effect on soil protein. I should also mention, while I'm on this slide, the samples were taken before the rice was planted, when the field and the mung bean had been harvested, when the field was fallow. So this is after the 3rd planting and before the next planting of rice is when these samples have been taken. All right, getting into, this is my same graph, so we will skip it. Okay, we're seeing aggregate stability. So one of the things, this is the only physical characteristic that we've been able to measure so far, given some of the data that we're collecting. And aggregate stability is up here in y-axis as % stable aggregates. And again, the blue is still rice, mustard, mung bean. Purple is rice, wheat, sorghum. And we're not actually seeing any differences between these plots. And these are all fairly small plots, so maybe that is in a way understandable, given that we're not seeing much effect of either of this. Okay, experiment 2 is looking at organic crop residues and inorganic fertilizers on rice, wheat systems. So you have 4 organic fertilizers, which is no manure, farmyard manure, crop residue, and a combination of crop residue and farmyard manure. And then you have 4 fertilizer treatments, which is 0, 50, 100, and 150% of the recommended rate. And the first thing we're seeing is that there's no effect of fertilizer or organic amendments on active carbon. There appears to be a trend going from no manure up to farmyard manure in the means of these treatments, but they're not significant. In addition to, but when you're looking at soil protein, we're now seeing the same differences. Again, no manure is, there you see the same trend. So soil protein going up from no manure systems up to farmyard manure and crop residue systems. And you can tell it's up here it's mostly CDs, and then up over here they're A's. There is a clear difference. We're not seeing a lot of difference of the recommended fertilizer rate, though. So again, in some places there is that expected, as you increase fertilizer, soil protein is going up a little, but that's not consistently true. Up over here, farmer manure and crop residue. the means show greater respiration. So if you look at the mean of no manure, farmyard manure, crop residue, these three aren't particularly different, but the farmyard manure plus crop residue mean is different. But we're not seeing differences, again, within each treatment for respiration. And again, we're not seeing significant differences in aggregate stability. So that's going to be true of all the experiments that I look at. And one of the reasons that could be, and I forgot to mention this in the first experiment, is all three experiments are aggressively tilled. So whether that's incorporating the crop residue or just planting, they're all tilled aggressively. So any effect that you might see of these treatments on aggregate stability is probably all mitigated by the tilling. Okay, the final experiment in the university plots is a long-term study on crop residue and zinc application. This time we have 4 organic amendments, which is, again, 0% crop residue incorporation, 25, 50, and then 100% crop residue incorporation. And then looking at 4 treatments of zinc, which varies from 0 to 10 kilograms per hectare. Okay, so looking at active carbon again, or labile carbon, what we are seeing is that, interestingly, with a 0% crop residue and 0 zinc, what you would imagine would be the lowest, lowest fertile soils actually shows one of the highest active carbons. So the 0% crop residue actually goes from high to continuously low. But beyond that, you do again see the same trend of going up as you go up the crop residue level increases, your active carbon also increases slightly. And the same trend in soil protein again, though this time without the oddness in the 0% crop residue level. So you see that trend going up from VC to up over A over here. Respiration values, we see a same trend, but not a significant difference in this one. Okay. And aggregate stability, again, we're not seeing some of the same differences. We're not seeing the differences that we were expecting. Okay, so getting into experiment 4, which is the one in the Patna plots, there are 4 different scenarios to this. So scenario 1 is general farmer practices. So this is what farmers are doing in that area itself. So it's rice conventionally tilled, wheat conventionally tilled, and the third season left fallow. Scenario 2 is the best management practice. So like just slightly tweaking the system. So you have rice conventionally tilled still, you make wheat 0 tilled, and you add mung bean, which is a legume, to the system. Scenario 3 is to deal with water scarcity. So in terms of, so to aid with water conservation, rice, wheat, and mung bean are all 0 tills now. And that also aids with labor as well, because that's a big issue in the state. And scenario 4, which is a more intensified, diversified system that might take a little while to get to, is you have rice 0 tilled, mustard 0 tilled, and a spring maize that's 0 tilled. And looking at the 4 scenarios, what we actually see is that the best management practices have the highest active carbon. And the best management practice, again, is like the rice conventionally tilled, wheat 0 tilled, and mung bean conventionally tilled, which is tied in first place, actually, for the water conservation. So that's where rice, wheat, mung bean are all 0 tilled, which is good news in terms of trying to move people away from the more conventionally tilled farmer practice. We also see that same trend in soil protein, though this time the variability was higher and we weren't able to find a significant difference. With soil respiration, what we see is, again, the best management practice does really well, as here does the intensified and diversified systems and farmer practices. So this is slightly interesting. One of the issues with respiration in India is also it's really hard to get a consistent temperature, because while over here all our labs are temperature controlled, that's not true of India. So respiration data is variable throughout this experiment, and that is tricky. All right, so coming up into some conclusions. We do see some of the expected trends that we were expecting in soil health, in like looking at the soil health data, But there are also some stuff that we're not really, that was unexpected and we can't explain just yet. That's laden by the fact that a lot of this is still preliminary data. So we're still getting information on fields management data. We're still getting information on yield. And once we have all of that, that should give us a better picture of what we're seeing because we can correlate it with the lab results. And to that, I'll be spending actually the next 8 months, starting from about June to the beginning of next semester in India, collecting and analyzing more data. Okay. So I'd like to thank Harold up over here, my committee, and he's up over here as well. Prabhu Bengali. Ranjan Laik is the professor who works at PUSA and has been helping with my research a lot. Bob in the Cornell, up in the soil health team, has been incredibly helpful with the data and the results. Santosh has been working in India analyzing some of this data. And Zach, also the soil health team, has been really helpful. This is actually us in India doing a soil health workshop last summer where we were going over some of these same indicators. And thank you. Okay, I rushed through, so there's a lot of time for questions now. Mm-hmm. Yeah, absolutely. So most of these are actually rice wheat systems because that's what's most common in Bihar. And the rice is generally sown the minute the monsoon hits. So it's a bit of a varying time. Summer I collected these, they were collected in the first week of July because the rains actually hit later that year. And we were trying to get it with the first rain and hits, the ground wasn't too hard to sample, but before obviously it rained enough that people would start planting the rice. That's what we were aiming for. Just because that's the best time for us to be able to get the sample out of the field as well, and also we won't have any interference with the crop. And the soils were taken 0 to 15 centimeters, 0 to 15, yeah. So for actually the Patna plots, they were just taken with a shovel, a composite of 10 samples from the field, and then they were all mixed in together and created up into one sample. The Pusa fields are actually like, the plots are much tinier, so we weren't able to get like a shovel and go in there because your field is only about as big as this. So those were more taken with shovels. There was still a composite of 10 samples around the fields. It's like in W pattern that we go through. Does that answer your question? Okay. All right. Yes. One of the reasons is mustard is grown a lot in the area as well, more as a cash crop. The more physiological reasons behind mustard, I actually am not sure about. So that's something that I'd have to look into more. But the reason that a lot of our university plots look at it is because they use mustard for mustard oil. So they use the seeds for oil, and it's a very common cash crop there. Yes.
It would have been helpful to see the error bars on those, as well as all the ABs and all that. Get some idea of variability. I was going to ask, it looked like there was some fairly obvious trends, but they weren't significant. I understand.
Yeah.
Why do you think there was so much? How many replications have you had in each case, and then why do you think it was so variable?
Sure. So I actually did have error bars in, and then at the last minute I took them out just because I was trying to keep the data as simple as possible for everyone. The respiration data, like I said, has been the one that's most variable. And the reason for that is we try, you're using respiration to get into the methodology for each indicator, but we're using an electrical conductivity meter to be able to then calculate respiration. And it's kind of calibrated to a specific temperature. And in India, it's impossible to keep anything at a specific temperature because there's a day where it like starts off at like 0 and then goes up to like 30 degrees. Yeah. Right, and that is something that we're working on. Where we're taking just the actual KOH data every day and like trying to come up with how we adjust that. And that, so right now this is not adjusted data in any way. This is just the data that I got. I am looking at being able to adjust the data compared to temperature. That was only the respiration. Right, so for the first experiment that I did at the university plot, that's 4 treatments and 6 reps. And for the others, there are 3 reps each for 8 treatments. Yeah, so Well, so there are a couple of reasons. With the university plot, one of them is just, it's a very small plot. Like, these are tiny plots that generally we would be like, you have to have bigger, because otherwise you're just going to get like spillover effect from the plot that's right next to you is one of the issues that I think we'd be seeing. 3 is also just a small number. Obviously, having more than 3 reps is better to decrease variability. Yeah, those are some of the big things in terms of why I would assume there's variability. Yeah. Yes, I saw your hand first, sorry.
Yeah, thanks. You mentioned the income disparity.
Yeah. Yeah. Yeah.
I'm wondering, in the long term, if it's possible, thought about either doing it yourself or outsourcing economic analysis, trying to quantify your inputs monetarily and how they relate to soil health, or if a soil health indicator says it's green, what does that actually mean in terms of potential impact?
No, that's really interesting. Thank you. So there's actually 2 parts of the project that I'm like very interested in, but have done no review on. So that's why I didn't want to put it up in the presentation. But one of that is looking at nutrient flows across the state. And so that's something where I'm like, are we exporting crop residue? Where is the carbon and nitrogen flowing within the state itself and then maybe the country? And the second part is, can we then take the soil health indicator and turn it into an economic benefit, whether for application in insurance, in an insurance scheme, or for application in like valuing the land, but just being able to say, if my soil health indicator is this much, that's an economic benefit of this much to the land itself, to the farmer. Those are both things that I'm interested in, but it's still like very much in the information gathering stage. So while I would like to do that, I have no idea if it's going to be possible. Yeah. But if you have ideas, please let me know. I think up over here. So the first experiment is 1985. The others are also 1980s, but the exact date are skipping me. So the PUSA plots are a couple of decades old. The CSA plot is 2010, so about 8 years, if I'm getting that correctly. And maybe I can correct this. I do not have yield data yet. So that's one of the things where I was mentioning. I'm hoping to get, so the yield data exists. It's just actually getting the yield data, because again, like I said, the plot's gone through multiple professors. So this data is kind of spread around the place, and even getting like last year's yield data is like a bit of a process. So I'm hoping to get that and then being able to correlate some of this information. I also think it'll be really helpful in like crop residue trials, where I'm like, well, how much of crop residue is actually going back into the field? Yeah. Sorry, say that again? Yeah, I'm really interested on that too. I think just from hearsay, the plots with 0 zinc are showing a yield decrease, and you can kind of, in one of the pictures, just see the little patchiness of the soil, and I'll get there eventually. But yeah, so you do see patches in the soil, and you can just kind of estimate yield data that way, but actual yield data I don't have yet. Yes, sorry.
Is 10 tons of farm manure realistic?
So for a lot of smallholder farmers, manure is actually one of the main ways that they fertilize. So because there is a lot more of the integrated livestock approach in India, you have usually cows that people will keep for dairy, and the manure has to go somewhere. So it's either used for fuel or it's used for manure in a lot of cases. There's also a lot of fertilizer application, because fertilizer is subsidized in India, but which is why there's an experiment looking at both organic amendment and fertilizer. So yeah, I would say 10 ton. I mean, I don't have actual farmer data yet, so like I don't know how much they would be applying, but I would say it's reasonable. Yeah. Right. Yes, Dave.
I was just going to ask about when you gave the soil, your earlier table of the soil health numbers, had you recalibrated that the 113 is that different storing functions, or using the Cornell storing functions?
Those storing functions were not changed. Yeah. I just ran them to the Cornell's. Well, so the best subset analysis, none of that is my data. So that's all Phil's data from Phil's publication. So As far as I know, and maybe Harold can correct me, the scoring function was not edited, and that's just what he got from there.
I'm on that paper, I should know.
Yeah.
So any last question or?
We're doing a
I
time.
think the timing was great. Let's
Yeah.
get Kavya on our hands.
This has been a production of Cornell University, on the web at cornell.edu.